My name is Adeosun. I'm from the Department of Microbiology, Adeleke University, Adeosun State, Nigeria. First, I'd like to thank the Beyond Science Initiative for availing me the opportunity to present at this year's International Online Conference. Thank you very much. I'll be presenting on the topic resistance pattern of Pseudomonas aeruginosa isolated from selected tertiary hospitals in Osun State, Nigeria. Introduction. Pseudomonas species is an ubiquitous and diverse genus of gram-negative bacteria that can be found in the soil, water, decaying vegetation, and in animals. Of all the Pseudomonas species that exist, Adeosun et al. 2015 has recorded that the most important and ubiquitous pathogenic Pseudomonas is Pseudomonas aeruginosa. It is medically significant and it has a high resistance to antibiotics that causes wide spectrum of opportunistic infections ranging from very minor infections to severe and deadly infections. Pseudomonas aeruginosa is best known for chronic lung infections among cystic fibrosis patients, and it also causes serious infections among immune-compromised patients, especially cancer patients, burn patients, catheterized patients, and other hospitalized individuals. Antimicrobial resistance occurs in this organism when it becomes resistant to the antimicrobial drugs that are used for treating the infections it causes. Antimicrobial resistance threatens the effective prevention and the treatment of an increasing range of infections, including blood poisoning, pneumonia, diarrhea, gonorrhea, tuberculosis, HIV/AIDS, malaria, among other infections. The aim of this study is to determine the antibiotic susceptibility pattern and to evaluate the multiple antibiotic resistance index of the clinical isolate of Pseudomonas aeruginosa from selected hospitals in Osun State. The objectives of the study are to identify Pseudomonas aeruginosa obtained from clinical samples using the conventional methods, to determine the antibiotic sensitivity profile of the identified clinical isolate of Pseudomonas aeruginosa, and to determine the multiple antibiotic resistance index of the isolates. Justification of the study. Researchers have reported an increase in antibiotic resistance, and over the years, multi-drug resistant microorganisms have evolved. Ojana et al. 2014 has recorded this. This has resulted into treatment failure and over time has led to an increase in morbidity and mortality. This has resulted into a massive increase in death rates and in diseased conditions. This brought about the introduction of the last line of defense of antibiotics, which are the carbapenem group of antibiotics. Unfortunately, Gold et al. 2017, among other researchers, have reported resistance to the carbapenem group of antibiotics, which are supposed to be the last line result of antibiotics for the treatment of these infections. It is therefore imperative to determine the multiple antibiotic resistance pattern and the index of Pseudomonas aeruginosa clinical isolates so as to pose therapeutic measures for the treatment of infections, for the effective treatment of infections caused by Pseudomonas aeruginosa. The materials and method employed in this study are: 1. First, the determination of a sample size. The sample size was determined using the Fisher's-based formula as described by Daniel 1999. And secondly, the study area was selected. 4 tertiary hospitals were selected in Osun State. These hospitals include Wesley Guard Hospital, Ilesha, Osun State, State Specialist Hospital, Ashibiawo, Osun State, Lautech Teaching Hospital, and OAUTHC, Ile-Ife, Osun State. After which the ethical approval was sought and obtained from Lautech Teaching Hospital, Ogbomosho, Osun State. The ethical approval protocol number is as displayed on the screen. After the ethical approval was sought, the clinical isolates were obtained from the tertiary hospitals, clinical isolate of Pseudomonas aeruginosa precisely. The pre-identified organisms were confirmed to be Pseudomonas aeruginosa in the lab using the conventional methods. Biochemical tests were carried out on these isolates to be sure they are really Pseudomonas aeruginosa. After which, the antibiotic sensitivity testing of the Pseudomonas aeruginosa isolate was carried out following the Clinical Laboratory Standard Institute CLSI Guidelines 2018. The turbidity of the inoculum was standardized using the 0.5 Macfarlane standard, and the results of the sensitivity test was categorized as sensitive, intermediate, or resistant following the CLSI 2018. After the successful antibiotic sensitivity testing was carried out, the multiple antibiotic resistance index of each of the isolates was carried out according to the formula described by Oshundiya et al. 2013. Its formula states that the multiple antibiotic resistance index is calculated following the method that says the total number of antibiotics to which a particular isolate is resistant divided by the total number of antibiotics that was tested against that isolate. Multiple antibiotic resistance index was determined by an index greater than 0.2. Here is a map of Osun State, Nigeria, showing the towns of isolate collection. Oshogbo is a town where an isolate was collected, also from Ilesha, Ife, and also here is a result showing the biochemical test for Pseudomonas aeruginosa isolate. The result revealed Pseudomonas aeruginosa is gram-negative, catalase positive, glucose positive. For the sugar fermentation, it was negative. For mannitol fermentation, it was negative. Lactose fermentation revealed a negative result also. Motility was positive. The organism was tested and was found to be mobile. Oxidase was also positive, and a citrate test was also positive. Here is a figure showing the site distribution of Pseudomonas aeruginosa within selected hospitals in Osun State. On the vertical axis, we have a number of isolates collected per site of infection. And on the horizontal axis, we have the location of isolate collection. From this figure, the highest number of isolates within the selected hospitals in Osun State were obtained from Wesley Guide Hospital, Ilesha, Osun State. And of course, the urine, the isolates was gotten, the highest number of isolates was obtained from urine sample from Wesley Guide Hospital, Ilesha, Osun State. Here is a figure showing the distribution of Pseudomonas aeruginosa with respect to the site of infection. Pseudomonas aeruginosa was obtained from wound swab, urine, sputum, ear swab, eye swab, blood, and others. But Pseudomonas aeruginosa, the highest number of Pseudomonas aeruginosa was obtained from the wound swab and urine with 33% each. Here is a plate showing the antibiotic sensitivity test results of Pseudomonas aeruginosa. 15 different antibiotics that were oxide products were tested against all isolates of Pseudomonas aeruginosa that were obtained. And of course, these 15 different antibiotics fell into 8 classes of antibiotics. These 15 are different disc concentration. This is a figure showing the antimicrobial susceptibility profile of Pseudomonas aeruginosa isolate. This figure revealed that the highest level of susceptibility of Pseudomonas aeruginosa isolate was to imipenem, as 26 out of 36 isolates showed high susceptibility to imipenem. However, the least effective antibiotics to Pseudomonas aeruginosa isolate from this study was penicillin, as 35 out of 36 isolates showed high resistance to penicillin. The statistical analysis was conducted, and the result, as determined by one-way ANOVA analysis of variance, now revealed that there was no statistically significant difference between the groups of antibiotics tested on Pseudomonas aeruginosa and the anatomical sites. The multiple antibiotic resistance index of the isolate was calculated. In total, 30, that is the 83.33% of 36 Pseudomonas aeruginosa isolates, were found to be multi-drug resistant as they had a multiple antibiotic resistance index greater than 0.2. Discussion. Antimicrobial resistance in Pseudomonas aeruginosa has obviously become a very serious global problem and a global threat. This is recorded by Adeosun et al. 2013. Results from the study are shown now that the highest occurrence of Pseudomonas aeruginosa from the different sites of collection were from urine and wound swab, each of them having 33.33%. In consonance with the results obtained from this study, high occurrence of Pseudomonas aeruginosa in urine has been previously reported by Trivedi et al. 2015 as well as Odumosho et al. 2012, where the percentage of Pseudomonas aeruginosa recovered from urine was also the highest from their study, followed by wound swab with 20.40% among the other sites of collection. Previous study by Olayinka 2002 have confirmed urine to be the leading source of Pseudomonas aeruginosa in hospital settings because most patients who undergo surgery, they tend to be catheterized. This is a procedure which has been shown to create inherent risk for infections caused by Pseudomonas aeruginosa. This study also showed an high occurrence of Pseudomonas aeruginosa in wound. From a study in Enugu and Ebonyi states of Nigeria, as described by Amadi et al. 2009, there was 64% occurrence of Pseudomonas aeruginosa in wound. This is an indication that Pseudomonas aeruginosa is truly implicated with wound infections. From this study also, isolates of Pseudomonas aeruginosa demonstrated highest level of susceptibility to imipenem, which is a member of the antibiotic class of carbapenems. It revealed 92.60% susceptibility. This is in tandem with the result obtained in studies carried out by Oshundiya et al. 2013 and Tasali et al. 2013, where the susceptibility rate to imipenem from the study was 94.15% and 100% respectively. However, the highest resistance rate of Pseudomonas aeruginosa in this study was observed for penicillin with 97.22%. The obviously visible high rate of resistance of Pseudomonas aeruginosa to the penicillin group of antibiotics, which include the ampicillin, penicillin, ticagrelin, can be due to the fact that penicillins are generally cheap, they are commonly available, they are affordable compared to some other groups of antibiotics, hence making infected individuals purchase them easily. An easy purchase of this drug, even without the doctor's prescription, has given rooms for individuals to misuse the antibiotics. Hence, they have developed antibiotic resistance to the drugs. Non-adherence to dosage regimen also often leads to antibiotic pressure, where organisms they devise means of resisting the effects of the antibiotics that are supposed to combat the infections they cause. Analysis of the multiple antibiotic resistance index of Pseudomonas aeruginosa isolate in the study showed that 83.33% of the 36 isolates demonstrated resistance to not less than 3 antibiotics out of the 15 antibiotics that were tested. This corroborates the results obtained from the study carried out by Oshundiya et al. 2013, where nearly all the isolates, 91.20%, had multiple antibiotic resistance index that were higher than 0.2. This is an indication that a lot of organisms, especially Pseudomonas aeruginosa isolates, the clinical isolate now to be specific, have developed resistance to virtually all of the available antibiotics. This is causing serious problems, serious threats, and causing a serious challenge to administer the right antibiotics that would actually combat this infection. It is an indication that multi-drug resistance is increasing rapidly in many parts of the world, and this is posing serious therapeutic dilemma. In vitro sensitivity of antibiotics is an important factor that should be seriously considered in selecting the antimicrobial agents for treating infections. This is as stated by Kuma 2013. In conclusion, there's a high prevalence rate of multiple antibiotic resistant Pseudomonas aeruginosa in the selected tertiary hospitals investigated in the study. Some of the isolates showed resistance to some antibiotics that belong to the carbapenem class, which are the last line of defense. However, from the study, imipenem still remains a beneficial antibiotic for the treatment of infections caused by Pseudomonas aerugiosa. Recommendations are: greater innovations and investments are required in research for development of new antimicrobial medicines, vaccines, and diagnostic tools. Also, government should implement policies to prohibit indiscriminate use and prescription of antibiotics and improve on the public enlightenment programs to stop drug abuse. People need to be enlightened on the detrimental effects drug abuse has, which supports antibiotic resistance. It is also of utmost importance that a consistent monitoring of antibiotic resistance be done, as it will assist in the appropriate selection of the empiric antibiotic treatments in the proper setting. Here are the selected references used in the study. Thank you so much for listening.